Hi, everybody. Thanks for having me back here, especially since it always feels like I'm coming back to my home community. So today I'll be talking about developing and vetting assays in preparation for a deep mutational scanning experiment. Before I dive in, I want to point out that I gave an even more general overview for the workshop portion of the symposium last year. And it's still up on YouTube at the CMAP SEGS channel, so go back and check it out if you feel like it. But in today's talk, I'll more specifically touch on aspects you should consider when deciding and vetting your assay prior to your deep mutational scan. And this includes the choice of model system as well as choice of measurement, and also key considerations, including deciding what phenotype you're capturing and whether it's really representative of the biology you're trying to study overall. I'll finish by talking about the importance of understanding how expression level may factor into the desired output of your experiment. And I'll put a pandemic spin on all this by talking about these aspects in the context of a possible deep mutational scan of ACE2 variants while studying SARS-CoV-2 mediated entry. So if you attended the talks yesterday from Tyler Starr or Eric Procco, you should be pretty familiar with the biology of ACE2-dependent SARS-CoV-2 entry, but I'll still recap it here. Briefly, the receptor binding domain from the viral spike protein binds to ACE2 on the cell surface. And this binding then licenses the viral spike to go through a proteolytic activation step to free the viral fusion peptide that spears the target cell membrane. If it's expressed, TMPRSS2 in the cell surface can perform this proteolytic cleavage, which then allows the virus to enter the cell from the plasma membrane. But if TMPRSS2 is not expressed, well, that's not necessarily a huge problem for the virus, as it can become endocytosed and then become processed by endosomal proteases like cathepsin L to free that fusion peptide and allow the entry process to proceed. Once inserted into the cell membrane, the spike protein folds back on themselves, causing the viral and cellular membranes to fuse, allowing the virus into the cell interior and letting it wreak havoc and multiply to infect new cells. For the purposes of today's talk, all you're really need to keep in mind is that initial step of spike ACE2 binding, since that's what we'll be focusing on today. So what host model should you choose to perform your study in? Though you generally want to relate everything back to human physiology, you obviously can't perform experiments in humans. And even other model animals like mice or ferrets are cumbersome and expensive. And unless you're trying to study things like transmission or pathophysiology, that's way overkill anyway. Even working with primary cells is oftentimes out of reach, since it's hard to perform the necessary manipulations and get large enough library members into those primary cell cultures. You can also go to the other extreme, which is focusing on the biochemical aspects of the interaction outside of its normal context. And while that is generally the most technologically feasible route, and there's probably the most options in terms of systems which you can use to tackle it in that manner, it may require much more validation on the back end to make sure that the things that you learn are relevant to the physiology and biology of what you're trying to make claims about. The actual optimal system is likely to be very problem and experiment specific, where the Goldilocks solution is balancing physiological relevance, ease and robustness of use, but also what systems you're most familiar with, since everything will go a lot easier and you'll be able to avoid pitfalls. In my case, I've often sought problems that seem to be best studied with human transformed cell lines like HEC293Ts. But this is a host-virus interaction, so we have to also consider the virus side as well. And going back to the schematics from before, there are multiple steps along this pathway that are commonly measured to characterize this biology. The common strategy is just focus on the biochemical interaction between the two key protein partners. And in a sense, both Eric Procco and Tyler Starr's works do that. But when studying viral entry, as long as you have the right tools, you can also simply use viral entry as that readout in an infection assay. And since this readout is furthest downstream, it can work in multiple different studies, such as assessing the details of the protease cleavage steps. On the other hand, different cell backgrounds may have different expression levels or kinetics of these intervening steps, which may make them potential confounders. I'll end up talking about both of these types of assays a little bit later in this talk. But when talking about infection assays, there are still multiple choices. And of course, the more or the most physiological route is using the actual virus. But unfortunately, it's also a really hard route, since you need access to a BSL-3 facility and have to be willing to perform the experiments strictly following approved protocols while wearing a respirator. Also, the genetic engineering tools for SARS-CoV-2 work is still limited, and there aren't many options in terms of convenient reporters or markers, and you mostly have to rely on quantitating cytopathic effects. If you want to study any of those intracellular steps of SARS-CoV-2 infection, well, I mean, using the actual virus is pretty much your only route. But as I mentioned earlier, the goal in this example is to study the SARS-CoV-2 entry process, and there are tricks for that. A very convenient trick is to use hybrid viruses called pseudoviruses. And in this case, this involves using a more manipulatable virus, which you then coat with the SARS-CoV-2 spike protein so that it enters cells using the same ACE2-dependent mechanism as the actual SARS-CoV-2. There are a few kinds, but my favorite is lentivirus-based, since they're single cycle, so there's little worry about safety. There's a ton of recombinant DNA tools with which you can manipulate them, and they're also roughly the same size and shape as SARS-CoV-2. So despite them looking completely different on the inside, they look pretty similar on the outside, and that makes them a pretty great proxy for studying the entry step of SARS-CoV-2 infection. So aside from biological differences, binding and infection readouts are also good examples of two contrasting types of measurements. So binding is more of a continuous output, and here the amount of binding directly correlates with the mean fluorescent intensity. In this case, the low binding sample exhibited roughly 10 times less fluorescence than the high binding sample, and this brought that signal down to essentially background levels. If you want to turn binding into a deep mutational scan, you then have to turn it into a countable statistic. And you can do that perhaps with a mixed population of cells with diverse binding properties by separating the cells based on fluorescence using fluorescence-activated cell sorting, or FACS. Alternatively, a pull-down method where strong binders are retained during a wash step may be a cheaper and easier scaling method, albeit that comes with inherent difficulties in tuning the stringency of that wash to be maximally informative and reproducible. In contrast, infection can be considered a binary readout because cells are either infected or uninfected, and that can be assessed by a reporter gene like GFP. Though in real life, fluorescence from the reporter gene may not be perfect, and you may still get some cells with intermediate fluorescence, though you can still use a simple threshold like the dotted line that I've shown you here to distinguish the infected cells over the uninfected. Notably, this is a counting statistic where a 10-fold reduction in infectivity means 10-fold fewer green cells. So fewer cells that are just as green, but are no longer in that right side of that plot. You can also use fluorescence-activated cell sorting, or FACS, to sort the green cells away from the dark cells, but it's kind of inefficient and kind of expensive. And you can instead choose to swap out the GFP with an antibiotic resistance marker and turn the assay into a survival selection. And as long as your antibiotic resistance marker is well expressed, it should be pretty simple to find a concentration that gives you clean data, where infected cells are robustly antibiotic resistant, whereas cells that are uninfected are very antibiotic sensitive and die off and disappear. So it looks like we've chosen the host model, the virus model, and the readout. So I'm ready to start my experiment, right? Wrong. Before committing to a large-scale experiment, you want to make sure that the assay conditions will allow you to distinguish differences in the biology that you're trying to study. And in this case, it's ACE2 variants, so it's imperative to test a handful of ACE2 variants of known or expected range of effects before actually starting to make your library. Existing literature is a great resource for this step, and this situation is aided by an old paper from Mike Farzan's group after that original SARS-CoV epidemic in 2001. So there in that paper, using an IP pull-down assay, they found that the ACE2 variants K31D and K353D, which are both highlighted in yellow in a figure I took out of that paper, these variants exhibited background levels of binding with the SARS-CoV spike. While the SARS-CoV spike receptor binding domain has a similar footprint on ACE2 as the SARS-CoV-2 RBD. And you can look at the structure and see that these residues, 31 and 353, sit right at that interface. So it makes sense structurally that mutating these lysines to aspartic acids could have pretty strong effects. Well, instead of an IP pull-down assay, we just grabbed the SARS-CoV-2 RBD superfolder GFP construct that Eric Proko posted on Adgene at some point last year, and created some supernatants expressing this protein and effectively stained our ACE2 expressing cells. And as you can tell, the K31D and K353D ACE2 variants really drop staining with the RBD superfolder GFP, so that recapitulated the published results pretty well. But if I take those same exact cells and infect with SARS-CoV or SARS-CoV-2 pseudovirus, we saw little to no effect. So cells that seem like they would have worked based on that previous assay had no effect when we actually did the assay that we wanted to do that more closely recapitulates the biology in question, at least for us. Well, we were using HEC293T landing pad cells that I had previously engineered to have pretty strong transgene expression. So we hypothesized that strong ACE2 expression could be conferring strong avidity effects that were masking defects in affinity created by those ACE2 variants, and thus wondered whether we could reveal affinity effects if we reduce the amount of cell surface ACE2. We thus reduced expression of ACE2 by 30 to 100-fold, regenerated those ACE2 variants, K31D and K353D, and reperformed the experiment. And now we started seeing half-log to full log reductions to infection caused by these changes to the ACE2 sequence. So clearly, the conditions that were working for the cell surface binding assay was not working for the pseudovirus infection experiment. And had we gone ahead too early and made the library in the wrong system, well, we would have wasted a ton of time and resources. But that was only 2 variants. So to fully vet the assay, we tested a panel of a little over 24 variants in pseudovirus infection assays. And this was enough to start making some comparisons between the high throughput ACE2 variant binding data that accompanied Eric Proko's science paper and our own pseudovirus infection data. So as you can see in this figure, there's a correlated, albeit very much nonlinear relationship between ACE2 variant effects on binding and entry, with that transparent gray line there being a hypothetical 1-to-1 relationship. Notably, a number of variants that bound the RBD 2-fold or greater had no enhancing effect on pseudovirus infection. And furthermore, variants that had 2-fold or greater reductions in binding could be anywhere in the range of infection. For example, this R357T variant bound roughly 2-fold worse than wild type, but reduced pseudovirus infection more than 10-fold. While on the other hand, the I21N variant also bound roughly 2-fold worse than wild type, but promoted even more pseudovirus infection than wild type. But this is single cycle pseudovirus infection, and is it even reflective of real virus infection? Well, so far, I think the answer ends up being a more or less yes, as we took these same cells and challenged them with the SARS-CoV-2 WA1 isolate from Seattle, and these results corresponded pretty well. So ACE2 variants that supported pseudovirus infection also supported real virus infection, as observed by the data points in the top right part of this plot. And ACE2 variants that did not support pseudovirus infection also didn't support real virus spread, as observed by the data points in the bottom left corner. Similar to the results with pseudovirus, we saw a similar lack of linear correlation between cell surface binding and real SARS-CoV-2 infection. in my opinion, really hammering home that fact that binding results don't necessarily translate directly to infection. I do want to make it very clear here that this is in no way a knock on Eric Procco's data or paper. Since he set out to use the binding data he generated to find variants of ACE2 that bound better to create a potential soluble therapeutic, and he was able to achieve that. But with the binding data already there, some readers may be tempted to extend that binding data to be indicative of how ACE2 variants, perhaps in people or observed in other species, may affect SARS-CoV-2 infection. And I hope I convinced you today that it really isn't quite that simple. As a final fun aside, I'll note that the relationship between expression level and assay output may end up more complex than you might initially appreciate. So here I have 3 levels of engineered ACE2 expression: low, medium, and high. Stating if cell surface ACE2 is pretty straightforward and largely reflects the intended amounts of ACE2 expression. Single cycle pseudovirus infection also largely corresponds, though the high level expression doesn't necessarily give the same fold increase as expected based solely on expression, perhaps since ACE2 is no longer limiting for viral entry by those high levels. Well, something that surprised us was when we saw a far more complex relationship with multi-cycle SARS-CoV-2 spreading assays, where sure, having a medium level of ACE2 was really important for infectivity and gave us a lot more spread than the low condition. But there was apparently such a thing as too much ACE2, as the high level actually supported less viral spread than that medium level. And while this is still just a preliminary observation, my guess is that while high ACE2 likely helps the virus get in all these assay setups, it may be disrupting how the virus gets out and spreads to other cells. And that's something we would only see in the multi-cycle spreading assay with the rotavirus. Again, unless you really know the system well, you may be in for some surprises. So while the examples I gave today were pretty specific, I think there are some general themes you should consider before planning and performing a deep mutational scan yourself. So be thoughtful about balancing physiological relevance with feasibility when trying to tackle your problem. And personally, I actually oftentimes like to flip the script on this and think about what problems exist that are best tackled with the systems and skills I either currently possess or really want to master in the near future. Make sure you adequately vet your assay by testing representative variants before committing to a library or assay format. And as a general rule, survival assays tend to scale better than reporter assays, but reporter assays do come with the added benefit of being easier to monitor along the way. Also, biology is complicated, and the expression format you initially choose may need to be adjusted. Ideally, you would understand the full range of the relationship between expression level and output before embarking on your DMS to give yourself the best chance of not falling into those problems. So to finish, I'd like to thank the people who contributed to the contents of today's talk. And this includes Nidhi and Sarah, who are members of my lab and performed much of the cloning and pseudovirus infection work. And also collaborators here at CASE, particularly Anna Bruchet and Vinicius, who did the BSL-3 work on the cells that we engineered. I also like to thank the entities that publicly shared data and tools. And this includes Eric Procco, who deposited his constructs into Adgene pretty early on in this pandemic, which helped myself and probably a bunch of other researchers. And as you can also tell from my talk, the public availability of his deep mutational scanning data was important for the comparisons I made between ACE2 binding through his assay and the results you get with pseudovirus infection with ACE2 variants. BI Resources was also a very useful public resource as they provided the isolate needed for the BSL-3 work we did here. And I'd also like to thank NIH and ID for support as I started up my lab in September 2019, half a year before we had to pivot during this pandemic and the lockdowns. Finally, I'll be hiring more people in 2021. So if you're interested in this kind of stuff that I was talking about today, well, I'm always interested in recruiting like-minded people. to expand DMS and MAVE type studies here in Ohio. So with that, I think I'll end here. And because it's on the shorter side, I believe there will be time for questions. So thank you for your attention.